Hi, it's Hermione.
Hi, it's Carolyn. We'll be showing you how the TRP operon works.
This is another type of gene expression in prokaryotes. This specific operon is called a repressible operon, meaning that it is naturally expressed until a small specific molecule binds to the repressor and connects to the operator, stopping transcription. We will specifically be focusing on a tryptophan operon in the E. coli bacterial cell. Tryptophan operon works in a low tryptophan environment where the cell needs to make more, so genes set up the enzymes to synthesize it. Again, as a refresher, eukaryotic cells very rarely contain operons. Operons comprise of PROG, a promoter, repressor, operator, and genes. The promoter is the region of DNA that RNA polymerase can bind to. The operator is like a switch that turns the operon on or off if there's no tryptophan present. Therefore, the repressor is activated. The repressor is a protein that prevents RNA polymerase from transcribing DNA, essentially turning off gene expression.
Here is the key we will be using as we go through this process. Gene regulation begins during the process of transcription. Transcription begins at the promoter, the site where RNA polymerase binds to the DNA. However, the RNA polymerase will first bind to the regulatory gene to create the repressor to turn the gene on or off. The yellow brick represents the operator that the repressor will bind onto. The blue bricks are the promoters. And the 5 TRP genes, A through E, are the genes that will code for each enzyme that is needed in the TRP synthesis process. Tryptophan, or TRP, are the blue beads, and they are needed in cells because they are necessary amino acids in protein synthesis. The TRP gene will code for the enzymes that will help synthesize tryptophan when the environment is not providing enough of it. The repressor is inactive, which will allow RNA polymerase to bind to the promoter and synthesize the enzymes that will make the tryptophan needed for E. coli.
So now the E. coli has synthesized the necessary tryptophan that the environment lacked. However, when there is a lot of tryptophan from the cell's environment, it will waste the cell's energy to create more. So, TRP is a co-repressor because it works with the repressor to prevent transcription. The TRP binds to the repressor that is made by the regulatory gene. The repressor is now active and able to bind to the operator and block RNA polymerase from synthesizing more TRP genes.